Hello and welcome everyone. I am Babett Baumeister, Assistant Professor, PG and Research Department of Zoology, the New College, Chennai. Today we are going to be looking at this topic, which is going to be a small overview on the organogenesis of the vulva in C. elegans or Caenorhabditis elegans. So before we begin the actual topic, we'll just look at a small introduction on the C. elegans. As you must be knowing, C. elegans is a nematode worm which has a structure like this, and more importantly, the worm is almost transparent, such that you are able to visualize what is going on inside the body of the worm. Secondly, we need to introduce what is a vulva. Vulva is an organ of the female reproductive system which acts as a connection between the inside to the external environment. It is a point where the eggs are laid and also the point where sperm enters the body. In case of C. elegans, you have two sexes, the male and the hermaphrodites. The vulva can be seen as an organ in the hermaphrodite C. elegans worms. The vulva is a mid-ventrally located organ. So as you can see, this is the ventral side having cells numbered P1P to P12P, and somewhere um, along the middle or the midway along the length of the worm, you'll see formation of the vulva. So to be precise, the vulva will be formed between P5P and P7P, and ultimately you'll see this is the structure of the vulva. If you can see, this structure is actually a tube-like structure which is called the vulva. So this, as I said earlier, will be the point from where eggs are laid, and this will also be the point where sperm enters the body of the hermaphrodite worms. And this formation of the vulva will be a stepwise process, and we'll be looking at this process right now. So firstly, in this step, in step A, what you're seeing is there is a gene, Hox gene called Lin39, which will actually induce the production of a receptor called lead-23 onto these cells. These cells from P3P all the way to P8P. So all of these 6 cells will be having the lead-23 receptor, and because of this receptor, they are called the vulval equivalence group or the vulval precursor cells, because all of these cells have the equal capability or the equal competence to become vulva because of this signaling. In the next step, what we see is a cell called the anchor cell, which is actually one of the cell of the gonad. A specialized gonad cell called the anchor cell is positioned in close proximity with P6P, and it releases an inducing signal in the form of Lin3. The signal is such that it induces the primary fate in P6P. The Lin3 signal is also received by P5P as well as P7P due to the proximity, whereas all of the other cells, this P3P, P4P and P6P do not receive lin 3 and they adopt a tertiary fate and ultimately fuse with the hypodermis. Here what happens is this becomes P6P becomes the primary VPC or the first degree VPC, and then once it has achieved the primary fate, the first degree fate, it is going to send out lateral inhibiting signals. You see these arrows, these are lateral inhibitory signals such that the neighboring cells, that is P5P and P7P, do not adopt the first degree fate and ultimately they become second degree. So you have first degree here, second degree and second degree here, and then ultimately once these cells have been transformed into their respective fates, they undergo further mitotic division. The primary VPC ultimately gives 8 cells, the secondary VPC ultimately gives 7 cells, and this secondary VPC ultimately gives 7 cells. So I have a total of 22 cells, 7, 8 and 7 again. So you have a total of 22 cells forming this vulva. This shape of the vulva is formed due to the 22 cells and the anchor cell invading in between to create a opening. This anchor cell that you see here is going to finally invade in between here and create a opening. This diagrammatic representation that you have seen right now was taken from this research paper. You can go through this research paper later on. I'll be providing the links of the research paper and all my references in the video. So this again is another diagrammatic representation of the same. This is anterior axis, posterior axis, dorsal ventral axis. So as I said, these are the ventrally located cells. Some papers have P3P to P8P and some papers have P4P to P8P. So it doesn't matter. All that matters is these cells are all equally competent. So as I told you, they have the equal competence. These are called vulval equivalence group, vulval precursor cells or the vulval competence group, because they have equal competence to become vulva. This happened because they have the receptor lead23 which is going to receive signal from anchor cell. As you can see at this stage, that is the mid L3 stage or the third larval stage, this anchor cell is going to induce production of lin 3 which is going to transform this into the primary VPC, this into the secondary VPC and this into secondary VPC again. So you see the VPCs, these VPCs P5P, P6P and P7P, that is the 3 VPCs undergoing division here. In the L3 stage, late L3 stage, they've gone through first division, then they go through a second division and finally they form a structure like this, a tubular structure which is called the vulva. If you can imagine, there is a hole going through here through this point there is a hole going through, which is a connection from the inside of the worm to the outside environment. So again, reference for this diagram was taken from this source. This source is again going to be mentioned in the description of the video. So to summarize the entire process, what we have seen so far, the activity of a hoax gene called lin39. Lin39 activity is going to produce lead-23 receptors on the vulval equivalence group or the VPCs. That is, these cells are going to have lead-23 proteins due to activity of lin39. And after the lin39, we have the induction by the AC, which is going to produce lin3. So here you see the AC, the anchor cell, is going to produce lin3, which is going to induce a production of primary VPC, secondary VPC, and again a secondary VPC. You have lateral inhibition through lin12. So if you see here, these arrows, they are denoting lateral inhibition by the production of lin12. So you have a lateral inhibition there, and following that you have cell division of the VPCs. So this step, after the induction by the anchor cell, they are going to go through cell division, and after going through cycles of cell division to form 22 cells in total, they are going to go through a process called tubulogenesis. This is the ultimate step. This is going to form the structure of the vulva. This is the morphogenesis of the vulva. Finally, this structure is going to be formed after the cell division has been completed. So this has been a short video on the overview of the process of organogenesis of vulva in C. elegans. Hope you have been able to understand this video. Thank you.